Hello again, everybody. How beautiful was that? I am very lucky that Dr. Wong was like 15 minutes away from my house. And the cool thing is, I met a lot of families as well that used to come and have dinner with us, which was beautiful. How are things going so far? You guys learning a lot? Everything's going okay? Sweet. We're just trying to get some presentations up and running, and these beautiful people will come and speak to you about all of the treatments that they're currently working on, moderated by Mike Kelly. His accent's not so bad, is it, right? All right, so I'm going to get off stage and stop talking, but I just wanted to make sure everybody was doing okay. Please let me know if you have any questions. I'm going to be around. We'll be right back. Stay tuned. How was that?
Good.
Thanks. And then I'm after they're done.
Okay. Thank you, everybody. I don't know how to follow that after we've just witnessed Brenda getting the award and everything that's going on. But the only way that I know how to do it is to talk about some good science. And I think that there's the way to settle us back down from the level of excitement of the last one. We have a gene therapy session that I introduced this morning and guided your thinking about what you should be looking for in here. I'm going to spend the first few minutes actually talking about what is gene therapy. And the whole purpose of that is so that each of the presenters coming up don't have to say the same slides and introduce that. So it's a way of getting us all set up so they can run straight into the data that they've got. So a handful of slides. This one here talks about the rationale for gene therapy. This is my first one. Yeah, gene therapy aims to replace a defective gene with a new functional copy. It is that simple. In Duchenne, we simply just don't have the benefit of being able to inject dystrophin back into your muscle. Unfortunately, it doesn't work that way. There's some diseases that you can actually do that, but with dystrophin, because it has to get well inside a muscle cell, we need a thing to carry it in there. So we take the gene and we put it inside something that carries it, and that's an AAV. It's an adeno-associated virus. We use that because it's something that is inherently quite safe. It targets muscle very efficiently. Viruses have been around for millions of years, if not billions of years, efficiently been putting their genetic material inside cells. So this is the preferred delivery method that we've got in order to put a dystrophin gene back into muscle. The benefits are, as we'd expect from an AAV, it's a single-dose gene therapy. You do it once and it's one and done. There's high transduction levels, so that simply means that the gene is efficiently delivered in and turned on. Broad specificity for lots of different types of tissue. So an AAV not only goes after skeletal muscle, cardiac muscle, diaphragm, does all that extremely efficiently, and that's why we use it. And the therapeutic benefits are long-lasting. The challenges are ones that's quite clear to everyone in the room. There is high doses required in order to target muscle. And if you think that muscle is about 50 to 60% the total mass of the body, you need an awful lot of this virus in order to really target all that there muscle. Because that's not the only tissue that an AAV goes to. It goes to the spleen, it goes to liver. But as I'll tell you, that we want it specifically for muscle. We don't have an ability to redose. Like any virus, once you're exposed to it, the body reacts in order to produce antibodies that are designed really to see if there's another infection. So this is the reason why it's a one and done. The antibodies that are produced from that initial injection, they stay around for many, many years. Pre-existing immunity is also an issue with an AAV. What that essentially means, AAV is a natural virus. It's in the environment. An individual can actually get contacted with it, and you don't get sick. It doesn't cause any disease in humans, but it leaves behind that fingerprint of neutralizing antibodies from an environmental exposure. So we reckon that around about 25% of individuals out there will have been exposed and will have antibodies, and that'll preclude them from getting gene therapy. And the final point in this slide is an important one, and we're going to deal with it as we get to the end of this session. It's limited cargo carrying capacity. AAVs, or particularly any sort of virus, there's a whole range out there that come from the very small to the very large. An AAV, an adeno-associated virus, is one of the very, very small ones, one of the smallest ones in sex. It's about 30 nanometers in size. Now, that may not mean a lot to you, but I'll contextualize it. 30 nanometers is about 1/2000th the diameter of a human hair. This is a very, very small virus, and therefore it's not a shock to find that we can't get a lot of carrying capacity inside that. I heard Liana say this morning that the DMD gene is the largest gene that we've got in the human genome, and it transcribes for a protein, dystrophin, that is incredibly large and much too large to fit inside an AAV. The cartoon at the top of the slide shows you the coding region for dystrophin. It's about 14 kb. And the carrying capacity of the gene is roughly about 4.7 kb. You need other things in there besides that, the promoter and various other components. So the actual coding region that we've got to play with is roughly about 4.5 kb. That's about a third the size of the normal gene. So we have to work hard in order to figure how do you get truncated and design truncated proteins that we're able to fit. And that there comes from, again, the cartoon at the top. You've got the full-length dystrophin there of roughly 14 kb, and a truncated form that came from an individual that has lost almost half of their coding region. That individual that had that there gene was quite normal. He had an asymptomatic disease. He walked with a cane whenever he was 61 years of age. He had a sibling that was a weightlifter in his 20s. So what it shows you is you can actually make really short truncated forms of this gene, and you can still have high functionality, an asymptomatic disease, and live a normal life. And that's the basis for each of the genes you're going to hear from the companies. They've all designed around this, even shorter forms than the one that you've seen there. And there's where we've got the ability now to actually package microdystrophin genes inside this AAV. The gene therapy products from each of the companies have got various things that are in common. Each of them have, and I've put them on this slide, this N-terminal actin binding domain, which binds deep within the muscle cell, tying down the dystrophin protein. It contains a number of the hinge regions that are part on this. This is the yellowy part of the cartoon. And a small number of the spectrin repeats that are in the center part of this, and a truncated C-terminus. And as I mentioned this morning, you can see Regenix with a slightly larger form. But there's differences between each of the four, and those differences are set out in this slide. There's 4 individual different serotypes that are being used by each of the companies. Sarepta is using an RH74. Insmed is using an AAV9. Solid is using a lab-modified one, SLB101 from AAV9, and that was modified to make it more targeted towards muscle and less targeted towards liver. And we'll see how that's going to play out with Regenix using AAV8. Each of them are using very specific muscle promoters, and it's important to understand what that is. This virus goes to a lot of places. It goes to the liver, the spleen, a lot of muscle groups, but we only want the gene turned on whenever it's in muscle. And these muscle-specific promoters do exactly that job. That's the way they work through so that the gene is only turned on in muscle and is present in there. And there's different types of promoters. The SPC512 promoter is slightly smaller, and that's allowed Regenix in order to make this slightly larger gene at an important point on the C-terminus. And Solid has modified their gene in order to include the nNOS binding site. So the way that I would want you to look at that is these are not all the same products. There is 4 individual shots in here that give us an ability to be meaningful and successful. And as these march through clinical trials, I'm expecting to see some differentiate from others. And that's what the trials will hopefully show over the next coming months and years. So I'm going to welcome out on stage each of the companies one at a time. The first one is going to be Sarepta, who's obviously, as you know, dosed more than 800 patients with ELEVIDYS. And there's a tremendous amount of information that we know about this molecule already. And there's more that's going to come out of these, followed these patients for quite a long period of time. They're now dosing very young individuals from 1 year upwards. That'll be followed by REGENXBIO to update us on the phase 3 portion of the Affinity trial that's going to fully recruit this year, and we expect to see top-line data. And I think you'll hear their plans to file for accelerated approval roughly at this point in next year. Solid Biosciences will be talking about their INSPIRE Duchenne trial that's recently continued to expand recruitment, and they're on track to release pivotal data later this year, and they'll come out and talk further about where they are. And Insmed with the ASCEND study that's just beginning to be recruited. And again, as I mentioned this morning, different from the others in that it's intrathecally administered, and they believe that there's a better target for the heart and for skeletal muscle. So with that, I'll step off stage and I'll invite Sarepta to come on and start talking from there. Wendy? There you go.
Good morning, everyone. My name's Wendy, and I lead patient affairs at Sarepta, which honestly means we have one of the best roles in the entire organization because we get to connect and listen and learn from families every single day. And it's our responsibility to make sure we bring your voices back into the organization. And it's through your feedback and all of the insights we've learned that we've been able to see the progress we have with the programs we've had at Sarepta. This presentation does contain some forward-looking statements. We're a public company, and so we definitely want to disclose that. Our commitment to Duchenne is across 3 technology platforms: exon skipping, gene therapy, and then we have some early research in gene editing. And you heard Paul and Deborah say this morning that their goal is to be able to treat 100% of the Duchenne patient population, and we share that goal. We want everyone to be successful in this space so that anyone that receives a diagnosis of Duchenne has a meaningful therapy. So I'm going to focus today on ELEVIDYS, Sarepta's gene therapy, which is the only approved gene therapy at this time, and just really focus on the fact that the choices of vector, promoter, and transgene really do determine the safety, expression, and function of gene therapies respectively. So each gene therapy is different. I had the opportunity to actually share with you, and you heard Mike speak about it a little bit, that there's just a significant amount of ongoing work and clinical trials and research with ELEVIDYS. And this slide just summarizes all of the current programs, including observational studies that are underway. Part of this is our commitment to continuing to understand Duchenne, the impact of gene therapy, and the differences between patient presentations and making sure the right patients are studied and understood for the right treatments. I do want to clarify and be really clear, and I'm actually going to read this because this slide is so important. What is ELEVIDYS? ELEVIDYS is a prescription gene therapy used to treat individuals at least 4 years old of age with Duchenne muscular dystrophy and is approved for non-ambulatory patients at least 4 years of age, all of whom have a confirmed mutation in the DMD gene. ELEVIDYS for the older non-ambulatory children is approved under accelerated approval. So you may have heard about this, and what does that mean? Accelerated approval is a pathway afforded in the US through exquisite FDA oversight and opinion and intervention that allows for approval of a drug to be based on a marker that is considered reasonably likely to predict clinical benefit. ELEVIDYS treatment increased the marker called ELEVIDYS microdystrophin in skeletal muscle. Verification of this clinical benefit may be needed for ELEVIDYS to continue to be approved for non-ambulatory patients with DMD. So this is ongoing work, and we're really excited about all the data and learnings that are being gathered. There are patients who are not eligible for ELEVIDYS who have certain types of mutations or deletions in the exon 8 or exon 9 DMD gene. And we do want to make sure that everyone's aware, if considering treatment with ELEVIDYS, that there's an understanding that there are things to know about the treatment. There are infusion-related reactions, all of which are listed here. And then ELEVIDYS can increase certain liver enzyme levels and cause acute serious liver injury. Patients who receive ELEVIDYS will be prescribed a high-dose corticosteroid medication, including before and after infusion, and people will undergo a series of blood tests to really monitor what's happening and ensure that any adverse events can be handled in the right manner. I mentioned that I have a really special privileged role in the company, getting to know families. And just a couple weeks ago, I met a mom named Cheryl, and she shared with us her son Charlie's experience, really her whole family's experience with ELEVIDYS. And we thought today it'd be really powerful for you to hear from her instead of me.
Duchenne can vary in presentation, severity, and rate of progression. This is one person's experience and may not be representative of all individuals with Duchenne. People who receive ELEVIDYS may have different experiences and varied treatment responses. Talk to your doctor about what may be right for you or your child.
My name is Cheryl. My husband Charles and I have 2 boys. My son Charlie is 8 years old and he is living with Duchenne muscular dystrophy. Charlie was treated as part of a clinical trial. Charlie has a wonderful personality. He's a happy boy. He's always smiling. He's very social. He loves his big brother Noah and playing with him. He's really smart. He loves math. He loves going to school. He loves to learn new things. He's constantly telling me things that I never knew about space or science.
Charlie is quite the kid. Charlie loves to explore the natural world. His mother and I are pretty dedicated to turning them into outdoor kids in a mostly indoor world.
Charlie was born a healthy child. As he grew, he started to miss some of his milestones. He didn't end up walking until 14 months. He fell a lot.
My reaction to Charlie's diagnosis was, quite simply put, it was just disbelief. It was a very emotional period for us, for his mother and I. Shortly after Charlie's diagnosis, we started the work on reaching out to a variety of resources just to wrap our heads around what it meant to be diagnosed with Duchenne and what options were available for treatment. Cheryl was really focused on connecting with families and understanding their experiences and how that might relate to us. A lot of my attention was paid to the kind of the education and the science, and what the clinical trials were doing, what the data meant. You know, in discussion with Charlie's neurologist, we began to understand the gene therapy candidate.
While we waited for Charlie to be treated with ELEVIDYS, we were really nervous. We were just anxious about it. You know, what was going to happen that day? What if we had a reaction? Just a lot of unknowns. We had a whole big bag of activities for him to do. We had Lego sets, we had books, we had drawing pads, markers, crayons, a little screen for him to watch a movie. We had all these things ready to go for Charlie. I was very nervous. I tried not to show it in front of Charlie. I acted really calm, but on the inside I was very nervous. When the whole thing finished and I saw that he was okay, I could just feel the stress lift away from my body and I felt like I could breathe.
It was a very happy day in our household. We did have a celebration which resulted in a very good meal that Charlie selected, if I recall. And we just celebrated the moment. And following treatment with ELEVIDYS, we felt confident we made the right decision for him.
After Charlie received ELEVIDYS, he started to show some improvements at home, and his teachers and aides and even his bus driver noticed some improvements as well.
My dream for Charlie is to have him accomplish everything that he wants to and be a supporter of his all along the way, no matter what the age, no matter what the circumstance, and I will do everything that I can to support him accomplishing whatever it is he seeks out to achieve.
Looking back, I would absolutely choose a Levitas for him again.
We've been fortunate enough now, being a few years with a Duchenne diagnosis, to connect with families, some virtual, some face-to-face, and we love telling Charlie's story. It's a rare disease. You know, the community in this is small, it's concise, it's tight, but you know, those who are involved in it are usually all in, and we're a strong part of that community. Not all families are going to have the same experience, and you need to expect that, but just be willing to dedicate the time and the commitment to learning everything that you can about the treatment that you're looking at, whether it be gene therapy or otherwise.
Duchenne has taught our family to live life to the fullest, to get out and do the things that we want to do as a family, make plans, have fun, just laugh and enjoy each other's company. My dream for Charlie is that he lives a full life and he does what he wants to do. He fulfills his dreams and makes the most of his life. I really just want Charlie to be happy and fulfilled.
So thanks to Charles, Cheryl, Charlie, and Noah, and I think we can all walk away remembering Charlie's dimples. Leading up to Levitas is a time of testing, decision-making, and determination. We really want to focus just for a minute on the important safety information again, that administration for Levitas may be delayed in patients who have acute liver conditions until the condition is resolved or under control, or anyone experiencing liver impairment. And there are immune-related myositis and myocarditis challenges that can be managed. And so for these reasons, it's really important that kids be as healthy as possible going into the infusion and then monitored very closely after the infusion, usually with weekly blood tests and other testing to just monitor what's happening. We know that the most critical time post-infusion is within that first 90 days. And then there is the need to follow a high-dose corticosteroid regimen, as I mentioned. So really monitoring that and being careful about reporting any adverse events is critical. We have all of this information in full detail at the booth here and also at levitas.com, and we are available to answer any questions. There are normal kind of important safety things to also remember, keeping up with vaccine schedules, and everything's listed here. And again, available at levitas.com, and we're here in the room as well, and we have a booth. To hear more stories, we heard Charlie's dad say, this is Charlie's experience. We have a number of other stories on the levitas.com website, which show different experiences, and we really encourage you to spend some time there. Mike's generously mentioned a number of times that over 800 patients have been treated with the Levitas worldwide. Why does this matter? It's a very well-studied gene therapy and a very well-researched gene therapy, with now also patients treated in what we call the real-world setting, so outside of a clinical trial. We're learning every single day, we being the scientific community and Sarepta and families, about the impact of this treatment, and all of those data, as you saw previously, are being presented in scientific forums, published and shared, so that we can advance the whole field. That's why this number matters. It's 800 boys that have been treated with Levitas to date. We remain committed to sharing all of this scientific learnings with the whole community. We encourage you to reach out to us directly in any of these ways, advocacy@sarepta.com, sareptassist.com, or levitas.com, and certainly here while we're all together at the Sarepta booth. Thank you so much.
So she's going to give it to me, okay?
On your way out.
Hopefully. All right. Thank you. Thank you. Hello, everyone. I'm Olivier Danos. I'm the Chief Scientific Officer at RegionX Bio, and I'm really excited to be here today to update you on the development of our gene therapy for Duchenne muscular dystrophy. our candidate is called RGX-202. So just a few words about our brand of gene therapy, our RGX-202. What you see here is a representation of the different vectors, AAV vectors, genomes that have been designed and are either on the market, as the Levitis you just heard about, or in clinical trials. Ours is the top one. So what we have in there is first a capsid, we're using AAV8, and we have a genome that encodes for a microdystrophin. As you see, it's pretty similar to all the other ones with details that are different. The main difference that we added is we've added more information for dystrophin into this microdystrophin, and that's represented in green. That's what we call the C-terminal domain. And the reason why we did that was that we knew from the literature and had also studied that ourselves, that having the C-terminal has a number of advantages, and some of them are summarized here, and this is from preclinical data in a mouse model of muscular dystrophy, of Duchenne muscular dystrophy, the MDX mouse. So what we see when we add the C-terminal to a microdystrophin construct is that we generate a protein that has a greater stability, it has a higher half-life in the muscle cell. That translates in the preclinical model by a higher potency and a higher ability to correct the muscular dystrophy. And this is shown on the diagram on the left here, where you see one of the assays that we've used that's called eccentric contraction. We generate in the whole muscle of a leg of the animal eccentric contraction, which are contractions that are also done while you're pulling on the muscle, and this is very lesional. This is the most you can do to a muscle to generate lesions. So what you see on this graph is the upper bar, the white dots, are measures of force after each of this contraction in a normal animal. And you see the normal animal is going to lose about 20% of its force after 20 contractions. An MDX animal shows a very dramatic loss of force, which is depicted here in gray. And in gray, you see that the MDX mouse loses about 80% of the force. When we come in with a microdystrophin, we do see the benefit, and we lose less force. But here we've compared a microdystrophin without the C-terminal in blue and with the C-terminal. And with the C-terminal, we always see a better performance. So this is just to illustrate to you the reason why we went all the way to add this information in the microdystrophy. So now, again, I'll pass quickly on that, but this is, again, an AAV8 vector that's meant to synthesize in the muscle cell the microdystrophin and allow for the formation of the dystrophin-associated complex and the recovery of muscle function. So our trial is called Affinity Duchenne. This is a phase 1/2/3 trial in boys with Duchenne. What I'm going to discuss with you today is the current data we have on the phase 1/2. We've started the pivotal trial. I'm not going to show you data. I'm just telling you that we've started and that we're including patients. But what about how did we come to this pivotal? Is through this phase 1/2 trial that we've run on boys age 1 to 12. And boys are obviously genetically confirmed for DMD. We exclude mutations that are in exon 18 and above for safety reasons initially. And we include only patients which are devoid of antibodies against AAV8. So we had a small group of patients, 3 patients that were dosed at a lower dose, and we observed the safety. Then we moved rather quickly to the second dose, which is our pivotal dose. We've now completed all that, and altogether we've treated 12 patients in this phase 1/2, and that's what I'm going to discuss with you now. So the demographics, several age groups. So we discussed the patient in several age groups because obviously you all know that the course of the disease is variable. There's a first phase where they still progress, then there's a plateau, and then it goes down. So whenever you're looking for improvement, you have to match what you're looking on your patient with an age match control, and that's what we're always doing in this in this analysis. What's showing here are these age groups that we're considering, 4 to 11 or 1 to 3. Now, safety, which is the primary endpoint of this phase 1/2, so far has been really satisfactory. We obviously observed the common drug-related adverse events, which are nausea and vomiting, fatigue, but we have a pretty clear picture when it comes to severe adverse event or adverse event of special interest. We have no myocarditis and no myositis. So it's a total of 12 patients, which is not a huge number, but it's good enough for us to be confident that we can move to a pivotal trial, and we conclude that as far as we've analyzed that, RGX-202 has been well tolerated. So what about the measure of gene transfer and the expression of microdystrophin? This is something we do at 3 months, 12 weeks. We take a biopsy in the treated patient, and we measure the amount of protein that's made by the vector and the number of copies of the DNA of the vector that we can find in there. Let's focus here on the right on the dose level 2. And again, here we have 3 groups, the 1 to 3, 2 to 4-year-old, 8 to 11. And throughout these different ages, we see a very good gene transfer with high expression of the protein and high copy numbers of the genome in there. So that's positive, but how does it translate to function? Again, this is an interim analysis. I'm not present. I mean, we're still working on the analysis of this functional function of these functions. What you see here are in blue the performances in either time function test or the North Star of treated patients. So the treated patients are in blue. The time function tests are time to stand, 10-meter walk run, time to climb. And we show the patients at the 2 dose levels, dose level 1 and dose level 2, which is again our pivotal phase. Dose level 1 has been now analyzed at 12 months and 9 months for dose level 2. In gray, you have the performances of age match controls. So you see that in every case here, we see improvement. So as far as we can tell right now, on this group of patients, we do have a positive trend in terms of function. Again, interim analysis, this needs to be completed, and data will accumulate and be released as we terminate this analysis. So what about the phase 3 that I told you was now open? Basically, it's the same kind of design. We're focusing on only 1 dose, which is this 2 times 10 to the 14 VG per kg. The pivotal endpoints now is different. We're going to look for the proportion of treated patients that can accumulate over 10% of microdystrophin in their muscle in the 3-month biopsy. And of course, we're going to study the secondary endpoints, which in a way are the most important, which are the function tests. So this is the design of the trial. Something I didn't mention is that we do immunosuppress the patients for the first 3 months, and this is tapered down afterwards, meaning that when we analyze function at 9 months or 12 months, immunosuppression is off. This is, and in the pivotal trial, patients will be observed over 24 months. So a number of sites, 6 on this map, are now open and recruiting patients. We're including more sites, so there'll be much more than that. We've already included some of the patients. It has started. We've dosed some of the patients. Obviously, you can find info on how to get in touch if you're interested in participating on our website. So again, this is a multi-center, open-label investigating RGX-202 as a potential for treatment of Duchenne muscular dystrophy. And don't hesitate to get in touch with us. Thank you so much.
Okay, hello everyone. Thank you very much to Mike, to Liana, to Deborah, and everyone at Cure Duchenne for, first of all, another amazing meeting and for this opportunity to present on our program. So my name is Pat Gonzalez. I'm the head of clinical science here at Solid Biosciences. I am the clinical lead for our SGT-003 program. And today I will be providing an update on where we are with this program and our ongoing clinical trial, which is the Inspire Duchenne study that Mike gave an overview of before. So as a quick update on kind of how things have been moving, I've had the privilege of being here at Cure Duchenne for the last few years. Last year, we gave an overview of what is the background of SGT-003. I'm very happy to today be able to give an update on the initial data from our study, and also additional updates on where we are and where we're expanding. We are a public company, so we do have a forward-looking statement slide. I'll quiz you on this later. And so I'll start by giving an overview of what is SGT-003. Both Mike and Liana did a great job of really emphasizing how each of the components of a gene therapy are unique. And as we're looking at a microdystrophin gene therapy specifically, even the microdystrophins are different. And so when we look at SGT-003, we have 2 major pieces that we want to call out here. The first is the microdystrophin itself. And so when we were looking at that full-length dystrophin, we know that it's a huge gene, so we need to miniaturize that and choose pieces to be able to fit within our AAV capsid. There are certain pieces of the dystrophin that are required. We know we need to maintain the structural stability of a muscle, so you need to form the links that are actually going to be on that membrane of the muscle, so that when you're contracting, it's protected. However, what we also included here was an additional binding domain in the center of the dystrophin that's called this nNOS binding domain. And so why does that matter? The reason it matters is because when you have exercise-induced demand, when you're using your muscles and you need more blood flow to get to them because you need more oxygen, you're going to need to be able to actually get what's called vasodilation, which is opening up the blood flow to those muscles. Nitric oxide, which is produced by this nNOS enzyme, is able to do that. And so when it's absent, when it's actually not localized to the membrane, this is something that your muscles are then more at risk of actually getting damaged because you don't have sufficient energy, you don't have a sufficient oxygenation. And so we're hopeful that this is something that could be additionally protective of muscle, more like that full-length dystrophin. So as we have this unique microdystrophin, we also have a unique way that we are delivering it. And so we talked about what AAVs are, and these are viruses that we're able to package with these microdystrophins. But what we did was go back to the lab and say, if we can actually try to optimize what already exists for naturally occurring viruses, we could be more targeted to muscle. And that means a couple of different things. One, we could be able to lower the dose. So from a safety perspective, we can actually get more expression with even less of a dose. And at the same time, if we're getting more to muscle and getting more expression, can we try to also limit off-target? And so when we think about off-target for AAVs, when you dose systemically, you get a lot to the liver. And so if you can try to reduce that, that could be ultimately very impactful for the safety profile of the drug. And so that's really what we saw as we started to work on this, this program and did non-clinical studies before getting into our clinic. And so here, when we just look at a monkey study that we ran, we can see that this profile of this new capsid does show multiple fold times higher levels getting to muscle. So here we're looking at both skeletal muscle and cardiac muscle, both critically important for Duchenne. And at the same time, we get about half to the liver. So this is really telling us that we're getting more where we need it and less where we don't. Really a positive profile for this capsid. We then wanted to actually look at microdystrophins and take the question of, if we're comparing this capsid to others that are being used right now, how will the expression levels compare? And so we did a head-to-head study with RH74, which is another naturally occurring capsid. And what we saw is that with SLB101, we do have about 4 to 6 times higher levels of expression when you administer with the exact same dose. And so this is something that really made us encouraged and made us move quickly into our clinical program for Duchenne. And so I mentioned the study here is called Inspire Duchenne, and it is a phase 1/2 first-in-human study of SGT-003. I'll start with really the data that we have to date, where we are, and then give an update on the study population and expansion that we also have ongoing, really excitingly as of this week. And so this is from a data cut that we had more recently, where we're doing a comprehensive evaluation of both safety and initial efficacy signs through biomarker analysis and muscle biopsy analysis. At this point, we had 7 patients who were dosed in the study. And so you can see here that they ranged in ages from 5 to 8 years old. Their weights here were up to about 28 kilos. And so those patients will give a safety update on, and the first 3 who reach the day 90 time point, which is where we collect muscle biopsies, will have muscle biopsy and biomarker analysis on as well. And we were very happy to see that in these initial patients who were dosed, we did not have any treatment emergent SAEs, so serious adverse events. None of these were reported. We also had no adverse events of special interest, where here we highlight them as looking at important things like hepatotoxicity, which is liver toxicity, liver injury, other things that could be more severe symptoms like thrombotic microangiopathy, myocarditis, myositis. We didn't see any of these in the study to date. What we did see were the more common things like nausea and vomiting, and all of those did resolve within those first couple of weeks post-dosing. So this was a very encouraging early profile. With that, as I mentioned, hepatotoxicity, that we did not observe any of this. I can also emphasize that we did not have any either biomarker or clinical evidence of liver injury. And when we look at one important marker of what we're looking at from a laboratory perspective, we really saw no changes in what's called GGT, which is what we would monitor very importantly post-dosing to look for any potential signs of liver injury. So seeing this flat throughout all of our patients who were enrolled in the study was a very encouraging sign, and hopefully potentially resulting because of the differences in the capsid. And now when we look importantly at the muscle biopsy results to really say, is the capsid getting where it's supposed to be getting? So is SGT-003 living up to what its potential had from the non-clinical? And this is very early data from the first 3 participants, but we were very encouraged to see that going from the left to the right, when we look at the left side here, we want to calculate how many copies are getting to muscle. This gives us an idea of, are we actually getting more than what we would have expected from a naturally occurring capsid? And the answer was yes. We saw that we had an average of 18.7 copies per nucleus in these muscle biopsies. And so if we think about a full muscle, 1 copy that will probably give you that one amount of protein for microdystrophin that can be made. Multiple copies not only allows you to have more of this production, but we're going to understand that there's some variability across muscles. So if you can have a higher total average copy number, you can account for that variability potentially to get more of that homogeneous expression throughout a muscle rather than heterogeneous expression. In the middle here really went from what are we seeing of number of copies to how is this resulting in microdystrophin expression. And so we use two different techniques here. One is Western blot that you may have heard about. Another is mass spectrometry. And these are ways for us to calculate how much microdystrophin are we making and compare it back to how much is a normal amount of microdystrophin. And by normal, we mean we take many different muscle samples from just normal donors, so healthy volunteers, who then we're going to be able to take an average of what would be the amount of microdystrophin or full-length dystrophin here that you would have in a muscle, and we compare that back. So we were very encouraged to see that in these first 3 participants, we had an average of 110% of normal, so really right around that normal percentage, if 100% is normal. In these muscles by Western blot, 108% of normal by mass spectrometry, so very consistent results between these 2 methods. And then for me, I'm a muscle biologist, physiologist, and I always like to say the more muscle we can protect, the more important that therapy is going to be. And so when we look at the right side, we use immunofluorescence as a way to be able to actually calculate how many different fibers throughout that muscle are expressing what we're calculating from this percentage of normal. And so we're very happy to see that we had 78% of positive fibers, which means not only are we making a lot of protein, but that protein is distributed throughout the muscle, which could be potentially important for that additional protection. We look at other downstream things as well to say, is our microdystrophin functioning on a molecular basis? So this is very important to actually look at where it is and then how it's impacting associated proteins. And so we can see that it is localized to the membrane. That's where it's supposed to be, like dystrophin is. And so baseline compared to day 90, that's where we see that the red here is dystrophin and microdystrophin. In the center, we looked at other dystrophin-associated protein complex members. This is beta-sarcoglycan, so it's decreased in a dystrophic muscle, and we can see that it's restored in microdystrophin-positive fibers. The same is true here with nNOS. So if we're looking at nNOS activity, which I mentioned is unique to this construct, that it's able to actually localize nNOS, we can see that that follows a very similar pattern where we are seeing the restoration of functionally and enzymatically active nNOS at the membrane, which is where it needs to be to have that potential effect on that increased blood flow to the muscle. We look at a lot of biomarkers too at this point because we're trying to look to see what can we have as early signs of what's the impact of expression on the stability of muscle, the integrity of muscle. And so I think everyone here knows CK very well. This is something that we see is increased in the circulation in Duchenne. And what we were happy to see here is that we had a substantial decrease, 57%, in this day 90 samples that we were analyzing. And when we look at other markers like that, when we're looking at LDH, AST, ALT, some of these are markers that you might think of that are associated with liver, but in Duchenne, they're also leaked out of muscle. And we were happy to see that across each of these markers, they showed a very similar pattern where they were all decreased quite consistently. Additionally, when we look back at the muscle, one important piece here called EMHC, which is a developmental form of myosin heavy chain, is telling us that the muscle is more stable. It's not turning over, it's not regenerating. So this could be another sign that our integrity of the muscle is improved when we have microdystrophin present. And then lastly, serum troponin is one thing that we look at from a cardiac perspective. And we were happy to do additional cardiac evaluations and say that in these early days, we know that the most important thing we look at for the heart is really for safety. We did not see any negative effects on the heart. We actually saw that there were some slight increases in ejection fraction and a decrease in one of our participants who had an elevated troponin I at baseline. This is something that fluctuates in Duchenne. You could see it increased. And we were very happy to see that that was an initial indicator that this could be having an additional positive effect on the heart. There will be a lot more that we're going to have to look at to be able to evaluate this further. But again, very positive that there were no decreases from a safety perspective especially. And so I gave an update of where our data are to date. And so what I also want to focus on is where this study is and where it's going, because we are newly expanding. And you can see the QR code here in the corner. And so I'll talk through this slide, but our clinicaltrials.gov posting has all of these details and will continue to be updated as we evolve the study as well. But what we've done is change this study from really being safety and tolerability to also including the primary endpoint of microdystrophin expression from an efficacy perspective. And so we're very happy now to not only make that change, but further expand the study. Previously, we had been enrolling patients who were 4 to 11 years of age. We have now also added a third cohort here that you can see on the bottom left-hand side, which is an under 4 years old cohort. So this is the first time that we're making the update that this cohort is now opening for enrollment. We just received our first IRB approvals for this week. And so sites will start to have their own local approvals as well. So if anyone were to contact sites, it may take some time for each individual one to come online with the protocol, but this will be opening up. And the rest of the criteria here are really going to be similar as what we had in the past. We do exclude deletion mutations specifically for exons 1 to 11 and 42 to 45. This is for that risk of a potential immune-mediated myositis, so a safety risk. We hope to learn more and to evolve this over time as well, but starting conservative, always focusing on safety up front. And then I'll point to the top left over here. We're also wanting to make the commitment to the community that we are looking at not only this population now from 11 and under, but also those who are older than 11 and those who are non-ambulatory. So we have included these additional cohorts in our protocol. They are not open for enrollment at this time, but as we learn more from these cohorts that are 11 and younger, we will then want to put safety data together, efficacy data, and then open these cohorts over time. So we'll continue to update the community as this study continues to evolve as well. And so we were very encouraged by the results that we saw. We saw the levels of expression that we were hoping for as a result of the capsid. We saw consistent results in these biomarkers, hopefully showing that these are initial signs of improved muscle integrity, and the safety profile that overall was positive and what we would expect for a gene therapy with no serious adverse events. I have to give a huge thank you to really everyone here, all of the families, all of the patients who are making all of this possible and really sacrificing so much to be able to be involved in this and move everything forward for the field and for everyone. Thank you to all of the teams. These are our clinical sites that are currently active as of today. We're continuing to expand. So we are now active in the US, Canada, and Italy. And more will be expanding, especially over the next few weeks. And so please go to our clinicaltrials.gov posting for additional information. And feel free to come up to any of us. We'll be in the exhibitor showcase and happy to answer more questions from there. Thank you all so much.
Good afternoon. My name is Brian Kaspar, Chief Scientific Officer of Insmed. We are so excited to be here at Cure Duchenne. The partnership that we have had at Insmed, how we've worked together over the years, is truly a collaborative experience, and we are so grateful for the support of Cure Duchenne, for the opportunity to be here, and to be presenting our ASCEND study trial that we are launching and is openly enrolling. So thank you so much for the opportunity to present. I do know that we are running late on the aspect of I am preventing you from lunch. So let me please present our research and development and our clinical trials. So thank you so much for the opportunity to present and to be part of the interaction yesterday, where we had clinical as well as FDA discussing how we develop new therapies for Duchenne muscular dystrophy. We're very happy to be part of the process and working together with many people who are advocating on behalf of the Duchenne muscular dystrophy field. So thank you for allowing me to present to you today. I would like to introduce the ASCEND study. This is the INS 1201. As Michael Kelly discussed earlier today, we are the new kids on the block. We have something new And our route of administration is different on the aspect of delivering better to muscle, potentially lowering dose to improve the safety profile of gene therapies. So this is a gene therapy study for Duchenne muscular dystrophy. What I also want to mention is I have a fantastic group of INSMED colleagues who are best in class, who are at the meeting, who are ready and able to answer questions and guide you to the INSMED resources that we have to answer questions and to guide you into the clinical trial operations that we have as part of the ASCEND study. I'll mention names here. Please look out for this fantastic group of individuals that I am really honored to be able to represent on this stage today. So in INSMED, we are a 1,600-person company global, but we're talking about Duchenne muscular dystrophy, and our mission is to transform the lives of patients with serious and rare diseases. And this really fits within our mantra of being able to attack these diseases from research and development with the best-in-class clinical development team and a clinical operations team that is going to be connected to the community to make your lives easier, for us to bring potential best-in-class therapies forward towards patients. We put patients first at INSMED, and it's easy to say, but it's hard to live. And I've been at this for over 20 plus years. I'm sorry, I hate to say those numbers because I'm aging myself, but we really take the patient experience really importantly as to how we deliver best-in-class therapies, safe-in-class therapies forward to the patients. We want to partner with you. We have been partnering with Cure Duchenne. We've got a great connection with this community, and we want to continue to advance on our understanding of the struggles within the Duchenne community and how we can best partner with you on our ability to advance drugs and therapies forward to this patient population. So Stephanie Duffy is our head of patient advocacy. You have me as Chief Scientific Officer speaking on behalf of INSMED. I've been at this over 20 plus years, and we have seen drugs to the market. We have seen patients' lives impacted by the power of gene therapy. We are pulling together all of our resources throughout INSMED to be able to make this a reality for the Duchenne muscular dystrophy patient population. So what is INS-1201? This is our first introduction to you as a community. We've been at this a long time. Let me just give a little background on what we've been doing. 4 years in development of trying to see some of the struggles and the challenges in the field and how we might be able to accomplish and tackle some of the problems to address safety, to address efficacy in the field. Now, these are preclinical studies. Let me just say, we are now enrolled, as you will hear, to address the patient population and enroll patients into our first-in-human clinical trials. But we're excited about our science. And it's based upon not only myself, but over 1,600 employees now as part of the INSMED network, as part of our global operations from Europe to Japan, to the United States, of developing best-in-class, first-in-class therapies to get that to patients. And that's what we're trying to do with INS-1201. Still early in development, but we have FDA approval to advance to human clinical trials based upon a robust package of preclinical data, safety, as well as a clinical trial design. You'll hear more about that. But what is INS-1201? It's a gene therapy in development for boys with Duchenne muscular dystrophy. So INS-1201 uses a well-characterized AAV9 capsid. We have a lot of experience with AAV9 capsids on thinking about the safety and where it goes, and is it targeting the tactical areas that we really need for this virus to get to deliver the gene. I liken this to a FedEx truck. We're delivering the gene exactly to the cells that need it most. And in Duchenne muscular dystrophy, that is skeletal muscle and it's cardiac muscle. We need to limit off-target effects, and that's exactly what we've developed with INS-1201. This is, again, AAV9 to deliver microdystrophin aimed at replacing the missing dystrophin and promoting muscle function and health. It will be administered as a one-time intrathecal. So intrathecal is into the spinal fluid as a one-time administration. It's a simple administration done by neurologists, done by interventional neurologists, intended to more efficiently target the muscle where it is needed: skeletal muscle, cardiac. We have a tremendous amount of preclinical data. This is an 8-minute talk. Again, lunch is right outside these doors, so I cannot spend the next 80 minutes talking what I would love to talk about of our preclinical data package. But what I can say from a scientist is that we have a strong preclinical package that is went to the FDA. I'll talk about that later. But this is one piece of data that says in a monkey, the closest that we can get to, so non-human primates are the closest that we can get to human beings. And what we can say is that when we deliver a gene, in this case that I'm presenting is a fluorescent gene, and we've stained for that fluorescent gene, are we targeting muscle efficiently? So what is important in Duchenne muscular dystrophy? It's tactical targeting of skeletal muscle and cardiac muscle. And we have, beyond reasonable doubt, shown in a dose-response method that we have a dose where we deliver intrathecally with a one-time dose that this targets skeletal muscles tremendously effectively. We also target cardiac muscle, again, extremely important for the Duchenne patient population. And one of the things that we found with this route of administration, remember Mike Kelly said, we're differentiated, we're a little bit different from others who are going systemically. The intrathecal route of administration lowers the dose for effective targeting to skeletal and cardiac muscle and avoids the liver and avoids other off-target effects. We believe, at least preclinically, that this increases the safety profile where lower doses, we target skeletal muscle, we target cardiac muscle more efficiently, and we remove the off-target effects. So we lower doses, we increase potential safety. Again, these are preclinical studies. This hasn't been tested in human beings, but we're excited about the opportunity to move these therapies forward. So, let me go back and say that we have our Executive Director of Research and Development on our Duchenne muscular dystrophy here at this meeting, Gretchen Thompson, Dr. Gretchen Thompson. She is fully committed to researching and developing in this disease state. And what I can tell you on our preclinical package is that we have been in the mouse model of the disease of Duchenne muscular dystrophy. And we have shown that there's a dose response, and we have shown profound correction of muscle health, muscle function, and improved physiology in our preclinical package. We've presented this to the FDA. Now, let me just say, a human being does not have a tail. What we do in the laboratory have tails, and those are the mouse studies. Let me just make a joke here. If a mouse ran across this stage, despite me touching them all the time and working with them, no one would jump higher than Brian Kaspar standing on stage. But this is our model to be able to understand whether we're targeting the right tissues and whether we are having functional benefit in the best models that we have. And that's what we have presented to the Food and Drug Administration. What we have also presented to the Food and Drug Administration is our safety profile. We have been in mice, and we have been in non-human primates with INS-1201 with a very clean safety profile that has allowed us the safe to proceed by the U.S. Food and Drug Administration. What we have now shown is we have shown that as we increase dystrophin or microdystrophin levels, that we have shown, Gretchen and her team have shown, that we have a functional benefit in these mouse models of the disease. Now, while I cannot proclaim that this is going to translate into humans, this is exactly what the purpose of a phase 1 clinical trial is for. To establish safety, we have profound preclinical data that says we should move forward. And that's exactly what we're doing. And I'm really excited about what we are doing at INSMED on launching the ASCEND study. So this is a multi-center study to establish safety. And let me just say, there is no placebo group. So everyone can take a sigh of relief right now. We are enrolling 2, 3, and 4-year-old ambulatory patients with a definitive DMD diagnosis. We're going to work on the older patients. Please stay tuned for more information from me and my team at INSMED. We're not leaving anyone left behind. Right now, this is 2, 3, and 4-year-old ambulatory patients. We have an independent data monitoring committee. We're being monitored. We're working very closely. We have the safe to proceed. Some of the tests at the study visits include physical exams, blood tests, heart activities, muscle biopsies. This is a needle biopsy from the principal investigators. So this is not an open muscle biopsy and biopsy and cutting into the muscle, a simple needle stick into the muscle. Physical function tests, growth and development tests, as well as questionnaires. Let me just say, before our lunch, where are we? This is where we are right now. We are opening 10 sites. Currently, we have 3 sites open, currently in Stanford, Arkansas, as well as the RDR Rare Disease Research Network. Many sites are very close to opening. These are fantastic sites. One of the things that I want to mention is that Insmed will work with you. If you don't see your site close to this, we may have new sites opening up. Please work with us, and please, I'm going to use 30 seconds to say, get out your cameras and take a picture of the code there, because that will give you more information on the ASCEND study, where our sites are, and how we can work with you. Not only will we help with travel at Insmed, but there are many resources from enrollment to site activations to site visits that our team will closely work with you to make your experience enrolling in our clinical trial as easy and safe and as much as we can do. So please reach out to us. We've got a great team here to discuss. So let me just, because they're telling me I'm out of time here, but our ASCEND study is exciting on the aspect that we're bringing a differentiated approach forward to the clinical trial. We have a safe to proceed from the US FDA. We're going to evaluate the safety and tolerability of INS-1201 by the intrathecal or spinal cord delivery, CSF delivery. We're enrolling currently 2, 3, and 4-year-olds. Sites are open, active, and more sites to come. So please seek out Samit Varma, our Vice President of Clinical Development and Gene Therapy here at Insmed. We have Mike Frazier in the audience, who has experience not only in the United States, but also launching drugs in Europe. Tremendous experience of gene therapy. We have Sharitha, part of our medical affairs, to answer questions and guide you into the right direction. Stephanie Duffy, to guide you into where you can answer your questions. We also have clinical operations and clinical here, Dr. Mark Stahl and Katie Church, who can guide you on our amazing connection to the Duchenne community, to PIs in our community, and our commitment to continue to advance and develop safe and tolerated as well as efficacious drugs for our patient population. Thank you so much for having me here. We look forward to answering questions.
Just leave that thing on. Thank you, Brian. Well done. Okay, everybody. Just join me in thanking once again their group that was involved in putting together the gene therapy one. A couple of things I want to say, and then there's one last topic before everybody departs for lunch. It reminds me, whenever you look and you're sitting listening to what each of the companies are talking about here, it's very clear that you can see the movement of these programs. You know, in the case of Sarepta, more than 800 patients dosed and is post-approval. In the case of Regenix and Solid, and now just the beginning of the Insmed, their advancement, is going to lead to, I expect, the next gene therapy product to be approved next year. So it is really, really good to see that their continued advance. The second thing I'll say is that each of them have talked about getting into younger and younger patients. And it really underpins the need for newborn screening. Being able to identify individuals at a very young age, because I think that's going to be an important part of the treatment options that are here. One last thing before I bring out our last speaker is really to talk about the breaking news session that we wanted. And I'm bringing out the CEO of Entos in a few seconds. Gene therapy has got an enormous amount of potential, and you heard that from each one of the speakers, but it certainly has clear limitations as well. The size of the gene, the inability to redose. And as the news that broke a couple of days ago was we completed the relationship and diligence and investment in a company called Entos that's got quite different technology that's involved in gene delivery. This The way that I'd introduced it to you, it's got components of it, like a virus gene delivery, and it's got components of it like lipid nanoparticles. For those of you that are not familiar with lipid nanoparticles, that's the formulation that was used for COVID. It's intramuscular injection into our arms in order to build up antibodies to COVID. It's a technology, as it speaks, lipid nanoparticles, that really target liver whenever it's given systemic. It targets the spleen, but it doesn't target any other main tissues. So it's got, even though it can carry large cargo and you can redose it, it does not transduce muscle at all. It can't target muscle. The technology that Entos has developed is somewhat of a hybrid between an LNP, a lipid nanoparticle, and a virus. And we believe that it's going to allow them to actually deliver large cargo large enough to deliver the full-length dystrophin gene. And it is targeted to muscle because of the viral-like nature of what it has. And that allows them to redose. There is no AAV, there is no antibodies, there's no restrictions for that. So we think that this technology is the next wave that's going to have an enormous impact in this disease. And armed with that, I'm going to invite out John Lewis. John is the CEO of Entos, who's going to talk for a few minutes about what this actually does. So John.
Thank you, sir. Yeah, hi everybody. I know lunch is waiting for us, but I just want to spend 5 minutes introducing you to Entos Pharmaceuticals and talking about just how wonderful it is to be here. I've had the chance to meet and get to know Deborah, Paul, and Håkan, and of course Mike and Leanna and the Cure Duchenne team, just a fantastic group. And just experiencing this conference today, I shed a couple tears already this morning, just remarkable stories. And I'm really excited to be a part of this journey. So Entos Pharmaceuticals is a genetic medicines company. We have a platform technology that is differentiated, as Mike said, from AAV and LNP. And we really see a huge promise for these new age genetic medicines because in essence, genetic medicines contain the instructions written in either DNA or RNA to have the patient manufacture their own treatments or cures for diseases. And so this opens up just a wide variety of potentially curable things that we already can do in a lab. So we can already reprogram T cells to fight things like cancer. We can already replace missing proteins in a dish. We can already activate or block specific disease genes. And of course, through gene editing, we can completely correct defective genes to potentially cure. And I keep saying in a dish because we can very easily do this in a dish, but it's much more difficult to do in a patient. And so, you know, the opportunity in this area is substantial, and delivery has really been the challenge. And so we've developed a platform called the Fusogenix PLV that addresses many of the limitations that Mike mentioned to be able to realize the potential of these life-saving technologies. Our platform is called the Fusogenix Proteolipid Vehicle. And as Mike said, it's sort of the best combination between some viral components. In our case, it's a fusion protein. And then lipid components similar to a lipid nanoparticle that almost all of us took during the pandemic. But what this combination allows us to do is enter the cell, introduce DNA or RNA into the cell in a completely new way. So by direct fusion, instead of having the cell sort of take it up and have it escaping, this directly fuses to the cell, and it does it in a very stealthy way, avoiding basically alarming the immune system and being able to get the DNA or RNA into the cell to have its curative intent. So as Mike mentioned, LNPs work really well for vaccines. But when you try to introduce genes through IV to the muscle, for instance, pretty much 85 to 90% of it is in the liver in the first few minutes. And this is because they're made with cholesterol, and cholesterol is actively trafficked to the liver. Because we have a fusion protein doing the delivery work in our platform, we're able to formulate with lipids or fats other than cholesterol, and that allows us to broadly send DNA or RNA throughout the body. And whether we deliver plasmid DNA or RNA, we're able to hit organs like the brain, liver, heart, spleen, lungs, and kidney. But I guess what you're really interested in is whether we can deliver to muscle. And we've done a number of studies. This is an example in a non-human primate where we investigated IV administration of a DNA-containing PLV and looked at sort of the organs it would go to. And we were actually pretty excited by these results. We see a pretty flat biodistribution across all the organs, but most importantly, heart is in there, skeletal muscle is in there. And this is with an unoptimized formulation that is not optimized for muscle delivery. Fortunately, we have the ability to pretty dramatically optimize these formulations for specific goals, and one of those is delivery to muscle. Because we don't need cholesterol for formulation, it gives us a lot of basically creative leeway to be able to develop formulations that deliver broadly beyond the liver, to have dramatically improved safety, to be able to be repeat dosed. And we've done repeat dosing studies in animals where we've dosed up to 19 times in a row. And most importantly for this, and the idea of being able to deliver full-length dystrophin, is the ability to encapsulate and deliver large cargoes. And there's really no limitation on the size of gene that we can encapsulate and deliver in this platform. Just want to give you an example that we just published late last year. It's a proof of concept study using a molecule called follistatin. Follistatin is really interesting. It's produced by your liver after you work out to help build muscle. So it helps build bigger, stronger muscles. And so as a proof of concept, we encoded human follistatin in DNA, encapsulated in a PLV, and gave a single dose to mice. And what we saw was pretty remarkable. After 6 months, these mice became jacked. They gained about 50% body weight, but it's all lean muscle mass. They roughly doubled their grip strength. So this is a great proof of concept that we can deliver muscle-building gene therapies, and the durability of this, the duration of this, is on the order of not days or weeks or months, but potentially years. Okay, so in our discussions with Cure Duchenne, and as Mike highlighted, there are limitations to the current approved therapies. And those main limitations are cargo size capacity. Ideally, we want to deliver the full-length functional dystrophin in these patients. We want to specifically deliver to skeletal muscle, cardiac muscle, and other muscles in the body. And we want to be able to redose. So ideally, either dose up to some functional level or to re-up the dose every year to make sure we have therapeutically relevant and potentially curative levels of dystrophin being expressed in muscle. And so we've been working over the past year to create a muscle-specific version of the PLV that I'm showing you a little bit of data for here. When we inject this into mice, we get really good distribution to all the skeletal muscle in the heart. We see, when we deliver this tool protein, we see it penetrate through all the skeletal muscles, and we can detect the protein in the muscle fibers in all of those muscles. So we expect, based on this, we'll be able to encapsulate DNA expressing full-length dystrophin and deliver it to all the skeletal muscles of the body. And that's really how I want to start today. It's my first introduction to the group here at Cure Duchenne and all of you wonderful people. We're starting this project right now. I want to introduce you for the first time to the NTOST ENT DMD 001 program. And it's basically building upon what I've just shown you. We have developed already a muscle-tropic PLV that distributes to skeletal muscle after IV administration. We see expression of protein throughout the muscle and in fibers. And we know from our large animal studies that the Fusogenics PLV is extremely well tolerated, can deliver large genes, and can be dosed repeatedly. So we're looking forward, with the support of Cure Duchenne, and hopefully many of you out there, to build the next generation of treatments for Duchenne. I really appreciate your time and attention, and happy to chat with you after and answer any questions.
Thank you so much. I want to see the jacked mice. I don't know about you guys, but I want to see the mice that are jacked. So I'm really not going to keep you from lunch. That was great. Thank you so much for all the speakers. The horizon is very hopeful for us right now. Thank you to all our industry partners, all of our panelists so far. There is a terrace outside to the right if you would like to eat outside in this very hot weather. Please come back at 1:30. We're doing a palliative care session here, and then there's other breakout sessions. If you look in your book, you'll see where you want to be. Enjoy your lunch, and we'll see you back soon. Thank you so much.
every potion. 'Cause if you like yourself a lot, don't let